Hello students. In the previous session, we have gone through cell wall and plasma membrane from the topic of eukaryotic cell. In today's session, we'll continue with the third aspect, that is endomembrane system. In the name itself, you can find the definition. Endo, that means inside. Inside the cell, there are certain membrane-bound cell organelles are present which are having coordinated function. Now such members together, as they are having a coordinated function, they are kept under endomembrane system. So what is endomembrane system? These are membrane-bound. These are membrane-bound cell organelles with coordinated function. Coordinated function. Now under this one, there are four cell organelles which we have to discuss. The first one, that is endoplasmic reticulum. Number 2, Golgi body. Number 3, lysosomes. And number 4, vacuole. We'll start with the first one, that is endoplasmic reticulum. Endoplasmic reticulum, that was first reported by Porter. Now these are network of tubular extensions which are coming from outer nuclear envelope and getting scattered into the cytoplasm. Sometimes it may reach up to plasma membrane also. So what are endoplasmic reticulum here? These are the network of, it's a network of tubular extensions which are coming from outer nuclear envelope and getting scattered into cytoplasm. Let's look onto the structure of endoplasmic reticulum. Endoplasmic reticulum, that consists of three parts. Number 1, that is cisternae. Now this cisternae, it's a reticular network-like structure which are having interconnected system. And number 2, it consists of a network of tubular extensions also, and these are sac-like extensions. They may be interconnected or else they may not be connected to each other. They are called tubules. And certain oval or spherical structures are present called as vesicles. Now, especially when it comes to cisternae, over the cisternae, it consists of certain pores. You can see here, it consists of certain pores, and these pores are called as porins. And these porins, they are due to a specific protein, and these proteins are called as glycoproteins. Glycoprotein. And the type of glycoproteins which are involved here, that is riboforin 1, and riboforin 2. Because of these porins, it acts like an attachment site for 80S ribosomes over the cisternae. And what is the subunit of ribosome that is exactly attached to these porins? That is larger subunit of 80S ribosome. So what is the function of porins? It acts like a site for the attachment of 60S subunit of 80S ribosomes. So if a question is asked, what is the subunit of ribosome that is exactly attached to the endoplasmic reticulum? That is 80S subunit, or else the larger subunit of 80S ribosomes. Now based on this attachment of ribosomes, endoplasmic reticulum texture will be of two types. It can be a rough texture, it can be a smooth ones. If it is rough, that is called rough endoplasmic reticulum. If it is smooth, that is smooth endoplasmic reticulum. So here, based on the attachment of ribosomes, endoplasmic reticulum is classified into two categories. Number 1, rough endoplasmic reticulum. Number 2, smooth endoplasmic reticulum. Number 1, rough endoplasmic reticulum. Rough endoplasmic reticulum, it is because of the attachment of ribosomes we said. But where these ribosomes are attached? They are not attached on entire part of endoplasmic reticulum. It is attached on much part of cisternae. So here it is some part. It is some part of cisternae. Then what about smooth endoplasmic reticulum? right? The rest of the part of cisternae. So here some part of cisternae. Along with that, the entire amount of tubules and vesicles. Now, what is the function of rough endoplasmic reticulum? As it is attached with ribosomes, they help in protein synthesis. It helps in protein synthesis. Now, this protein, it can get modified and helps in formation of enzymes. It means for the formation of enzymes, proteins are required, and these proteins, they are acting like a raw material for the synthesis of protein. So if a question is given, what is the type of endoplasmic reticulum that is responsible for the synthesis of precursor of enzymes? That is rough endoplasmic reticulum. So it helps in synthesis of precursor for enzymes, for enzyme synthesis. Coming on to smooth endoplasmic reticulum. Now, smooth endoplasmic reticulum, it helps in synthesis of lipids, and in case of animal cells, that helps in steroid synthesis. It can be rich in muscle cells, and in the muscle cells, whatever this smooth endoplasmic reticulum is present, that is called as sarcoplasmic reticulum. Sarcoplasmic reticulum. Now, whatever this smooth endoplasmic reticulum is present, it helps in uptake and release of calcium during the time of muscle contraction, right? During muscle contraction by uptake and release of calcium. It also secretes an enzyme which is called as cytochrome P450. Now this enzyme, it helps in detoxification of toxins or else foreign substances. So here it helps in detoxification of foreign substances or else xenobiotics, or foreign substance also you can say. Now apart from all this, now this smooth endoplasmic reticulum, when it gets detached and helps in formation of Golgi bodies. Now these are the functions of rough endoplasmic reticulum and smooth endoplasmic reticulum individually. But in overall, the endoplasmic reticulum that helps in two different functions. So let's talk about the overall function. Number one, as it's a network-like structure which is moving from the center to the peripheral side till the plasma membrane, it helps in forming cytoskeleton and helps in maintaining shape of the cell. Number two, it divides cell into two compartments. The inside region of endoplasmic reticulum, that is called as luminal compartment. In the diagram you can see. And the outside region, that is called as extraluminal compartment. So what is that? Inside, that is called as luminal compartment, and outside, that is called extraluminal. Now here we got a connection. Smooth endoplasmic reticulum, when it gets detached, that helps in formation of Golgi body, we said. So we'll talk about Golgi body.